All right, I think we can probably start with the seminar now. It's 2:02 p.m. our time. We still might have a few more people trickle in, but quick announcement. I just want to welcome everybody to this, what is it, June installment of our TACCster seminar series. TACCster, of course, is the TAC Symposium for Texas researchers, and this is a platform we use to have scientists, engineers, scholars, and students from across the state of Texas showcase their ongoing research that's using our TACS resources. We have a yearly symposium every September. This year it's coming up, it's September 23rd to 24th. It's going to be a hybrid format with in-person and online components. So please stay tuned to the TACCster website for more information and registration coming soon. Registration will be free this year, and I think it's opening next week. And if I could ask maybe Shweta or someone to just please post a link to the TACCster website in Zoom chat, that would be very helpful. Thank you. But today, so this is my absolute pleasure to introduce Dr. Lela Vukovic. She joins us from UT El Paso. She is an assistant professor in the Department of Chemistry and Biochemistry, and her lab's work, and I'm maybe interpreting this correctly or incorrectly, and you can correct me as much as you like, but my read of it is that they're using computational methods to advance the applications of nucleic acids in biology and in nanotechnology, and they're using them also to design molecules with high affinity target binding. So I think this research is very cool. Her talk today is titled Computational Modeling of Nanomaterials for Biomedical Applications, and I'm very eager to hear and learn and see what you have to show us. So with that, I will turn it over to you, and please feel free to share your screen. And Dr. Vukovic has also kindly offered to let people unmute, ask questions during if you feel the need, or post questions in Zoom chat, or wait till the end, however you prefer.
Thank you. Thank you, Joe. So thank you for the invitation. Do you see my screen?
Yeah, we see slides, but they're just in the PowerPoint mode.
Yes, I will just put it. Very good. Let me just.
Yep, that looks perfect.
Yeah. Okay. Is there? All right. Okay, it looks like. So I will then start my talk. Thank you for the invitation again to speak. We have really been tremendously assisted with the TAC resources in our research. And all of that I present today has been enabled with the resources from TAC. So as Joe mentioned, so I'm a computational chemist, and my group works in computational modeling of molecules, and we work at this interface of nanomaterials and biomolecules. We collaborate a lot with experimental groups. We try to interpret their result, help assist in the designing of new materials for interesting biomedical applications. So a lot of what I present, it will partly include the experimental results, and then what we did in what we did on our side in computation to help them interpret the data, or to help them with some new designs of what else they could try new that has some potential to be more effective for the applications. So here are examples of the kinds of systems we work with. So all of these are molecules. I guess most of you are familiar with some chemistry. So you can see here we have some nanotubes, we have some rings, molecular rings that you can see here. So you can see that we are working not on a small molecule level, but we are working on these nanometer scale sizes of systems. And they're sometimes materials, sometimes they're biomolecules, they're proteins, things like that. And the primary technique that we use for most of the research is called molecular dynamic simulations. Is there a way to move this? Yeah, okay, yeah, no, it's better. Okay, so it's molecular dynamic simulation. So what it is, it was shown here in a movie. It's basically a methodology where you take a system such as, you can see here, two proteins interacting with a nanoparticles covered with ligands. And then you describe in this system every atom that is making this system. You define in a physics-based models what interactions these atoms have with each other, and then you allow that system to evolve over time so that you get this type of trajectory showing you what is the system doing, what does it prefer to do, and as the system moves towards its hopefully lowest energy state. That's what the technique is, the primary technique that we use. So molecular dynamic simulations, it's giving you basically a movie of what the system made of the components you're interested in is doing on a pretty short time scale, such as nanoseconds, microseconds. That's what's available today with the resources. So that's what we use TAC for most of the time. So it has atomistic resolution, and it shows you how the system behaves. Oh, sorry, it looks like the slides. Oh yeah, here it is. So you can see what this works. The nanoparticle kind of goes from the top, it starts binding down. And that's what the technique gives you. The two projects I will talk to you about today are related, first of all, to how we can use nanoparticles to act as antiviral therapeutics, as antiviral agents. And I will show you what we did in that project with experimentalists together. So somebody tested these experimentally. And also I'll tell you in the second part briefly about design of nanotube-based materials as optical sensors of molecules that are called neurotransmitters. So I will tell you also about that. So in the first project, so with the pandemic, I think everybody's very much aware of the danger that the viruses pose to the whole society. And so in addition to coronavirus and the scheme and the images that are shown here, there is a number of other viruses that are out there such as hepatitis, HIV, herpes, HPV, human papillomavirus, all kinds of respiratory viruses are out there. And it's clear that we are trying to fight these viruses, and we are constantly trying to figure out the new therapeutics that are going to help first of all prevent infection, or to act therapeutically once the infection happens to reduce the symptoms. And typically the way that one designs such and prepares such therapeutics is that one has a target such as HIV, and then one tries to find a good therapeutic that will fight that particular virus. But since there are so many viruses out there, another strategy to find therapeutics could be different in nature. We could find some therapeutics that are broad spectrum, meaning therapeutics that can simultaneously be acting against a number of different viruses. So that's one of the strategies in finding the therapeutics, and that's what we have been working on in this project. So the way to look for molecules, materials that are broad spectrum, that can simultaneously then work against multiple viruses at the same time, is to look at what is shared in the mechanism, what is shared across these different viruses. And one of the shared things across different viruses is actually the initial mechanism of infection. So for example, HPV, herpes, and some other viruses, all of them start the infection in the same way. And this infection mechanism is shown here in this scheme. So for example, the first thing that happens when the virus attacks the host and starts the infection process is that the virus comes to the cell surface of the host, and there it kind of non-specifically binds to the molecules that are found on the cell surface. And those molecules are called heparin sulfates. We'll see what those molecules look like. And this initial attachment leads to the downstream infection processes. So, and many viruses, as I mentioned, share this initial stage of the infection. They come to the host cell, they initially bind to the heparin sulfate, and then all kinds of events proceed from that point on. So if we want to find broad spectrum therapeutics that would act against viruses which start the infection in this way, we could kind of utilize this mechanism and try to prevent it at this stage. If we can find some material that will prevent this binding, then we may be targeting multiple viruses simultaneously. So that has been the strategy in this project. So what are these heparin sulfates? They were just little red threads, as you can see here. But on a molecular level, this is what they look like. They're basically like repeating units of these rings. They're sugar molecules, repeating units. And on them, they have these sulfate groups that you see here circled. They're these functional groups that have negative charge on them. That's what they are. So that's what these sugars look like. And so what viruses do is when they come to the host cell, there are these types of molecules sitting on the host cell surface and they bind to them. So if we want to prevent this, and if we want to use this type of interactions of viruses with such molecules, we could use this strategy. The virus simultaneously probably binds with many of these functional groups that we see here because they're just repeating units on the cell surface. So in this project, what we have done is we have worked together with experimentalists to design and understand materials that are based on nanoparticles coated with material that mimic these heparin sulfates. Okay, and the idea of the nanoparticles is that the question that was posed is, can we design some nanomaterials that are easy to synthesize in the lab, which if you expose them to the virus, these nanoparticles coated with ligands that are similar to those sugars, if these nanoparticles bind to the virus, they may induce some kind of deformation of the virus, which will lead to inactivation of the virus. So that was the idea. Can we design such materials? And if yes, what are the criteria to help us design such materials? Can we show like a proof of concept, first of all, and then can we optimize such materials for this type of activity? And we worked on this with Francesco Stellacci's lab at Lausanne. And the short answer is yes, that's the case. So the experimentalists, they design multiple nanoparticles. So the nanoparticles have like solid cores and are coated with many ligands on the surface. And the cores were metal, gold, or iron oxide of different sizes. And then the ligands, they had different types of chemistries. So here you can see there are some ligands which have like 9 repeating carbon atoms, so they're pretty long, with this sulfonate group, so not sulfate but sulfonate group, which also has the negative charge and resembles the heparin sulfate. So this is what this type of chemical group is supposed to bind to the virus, since it's known that virus anyways recognizes similar chemical groups of this kind. And then other types of ligands were also tested. Some were kind of shorter, in the shorter ligands with the same group. Some had the different end groups, such as phosphonate. Some had a little bit polar chains instead of just the carbon alkane chains, etc. And the experiments showed that these types of ligands shown here, which had this sulfonate group at the end and pretty long ligands of like 9 carbon atoms, when you expose the viruses to these nanoparticles, which we label by MUSOT, that's just a type of ligand, MUS ligand, MUS ligand. When you expose the viruses in experiments, such a virus such as human papilloma virus, to the nanoparticles, they stop being infectious. The viruses stop being infectious. And when one looks at the microscopy images of what do these nanoparticles do to the viruses, after 2 hours, you see that the viruses kind of break apart when exposed to the nanoparticles. And that was shown for HPV, and then also human from this herpes simplex virus was also shown that it just falls apart. So such materials can be prepared. This was basically a proof of principle. Yes, this is how you can prepare the nanomaterials. And the question is, how do they work? And are there some hints that we can get from computational modeling to help us design new, better materials and understand how this happens, how this breaking of the capsid happens, what is the mechanism. So that's what we did. First of all, we looked at the human papilloma virus. Previously, people have resolved the structures of these viruses, such as human papilloma virus type 16. So we could actually just take the structure of this virus, viral proteins. We could model the nanoparticles and just see in such trajectories that I've shown you, how do these viral proteins and nanoparticles interact with each other. And we can just put different ligands on the nanoparticles and then track what happens in such systems. So that's what we did. You can see here the small 2.5 nanometer core nanoparticle interacting with one virus protein from the papilloma virus. And so we could see the way it is interacting, and we could also look at different ligands, which is what we did. What is it about the ligand that was destroying the virus that's different on a molecular level from the other ligands tested that were not destroying the virus, such as these MES ligands, which were much shorter, and the MUS ligands, which were destroying the virus, they were much longer on the nanoparticles. So when we look at it on the molecular level, we can see here that there is this viral protein, there is a nanoparticle with these MUS ligands. What we observed is, because the ligands are pretty long, these 9 carbon atoms, the nanoparticle is able to bind to the viral protein and make many contacts, make this multivalent binding. Because the ligands are long, they can kind of spread on the protein surface and make these contacts. And we, in addition to the charge binding that is the basis of recognition here, we also observed that the non-polar interactions of these long carbon chains are also important to stabilize the virus nanoparticle binding. When it came to these other ligands, such as MES, that experimentally is also tested and that didn't show antiviral activity, we found that because the ligands were so short, they couldn't make many interactions with the virus proteins. They were basically very rigid spheres, these nanoparticles with short ligands. So the length of the chain definitely played a big role, and also having ligand chains that can exhibit nonpolar interactions was also an important part of the mechanism. So then the question is, there were these microscopy images showing that these nanoparticles destroy the virus capsids, not just a single protein, but virus capsids. So we wanted to look what could be this disintegration mechanism. Why do the nanoparticles do this disintegration of viruses? And we proposed 2 mechanisms. One mechanism would be that the nanoparticle binds to a single protein of the capsid surface and then starts distorting that protein. So it's like an action on a single protein level. So that mechanism we call mechanism A. The second mechanism would be that a nanoparticle binds to that capsid surface, which we saw how it looked like, and then it starts distorting the proteins, the capsid proteins, their contacts, their junctions, and in that way destroy the virus. So nanoparticles could be acting with mechanism A, distorting single proteins, or distorting full capsids while preserving the viral proteins intact. So those were the 2 mechanisms which we then tested in simulations. And that's where a lot of the time went, because these are pretty big systems. So we first looked at this mechanism A. Is the nanoparticle destroying the virus protein itself? Is it kind of unfolding it or doing something to the protein itself? So the way to examine this mechanism is to track what is happening to the protein in these trajectories. and what happens to the protein when there is nanoparticle present and when there isn't a nanoparticle present. So there is a way to track the deformation of a protein using this quantity called RMSD. It's just a deformation, deviation. It's a root mean square deviation of the coordinates of the protein atoms. And you can see that over time, this deviation of atoms, it looks pretty similar when you have nanoparticle there or when you don't have it. So the mechanism A is not the correct mechanism. That's not how these antiviral particles destroy the viruses. It's not on a single protein level. We then proceeded to examine this mechanism B. If a nanoparticle binds to multiple viral proteins, is that leading to a distortion? The question is that leading to a distortion of the capsids? And is it going to break the capsids? So the student in my group, Parth, he just defended his PhD, he examined this mechanism B, and we looked at these different segments of viral proteins, 2, 3, and 4 protein segments, and we exposed these segments to nanoparticles and then checked what happens over time. Is the nanoparticle presence going to lead to disassembly of these capsid segments? And that's what we observed, in fact, in simulations. Here is the summary, very simple summary of what happened in them. When you have these viral proteins, the nanoparticles, first of all, what we observed, the nanoparticle binds at these junctions of 2 proteins, and you have 2 of them. And then we could quantify what the nanoparticle does. It actually increases the distance between these viral proteins. When the nanoparticle is present, the distance between viral protein increases. We could track it over time, as shown here. And also it increases the angle between the viral proteins. So we could also track that over time. So we could exactly see how the nanoparticle does that in these capsid segments. And also what we observed, we tracked the contacts that the protein form with each other. And when the nanoparticle is present, these contacts start breaking apart. The nanoparticle leads to the breaking of the contacts. So here we track over time the contacts between protein 1 and protein 2. And you can see, so the orange color signifies that there is a contact between a certain amino acid of these proteins. And you can see that over time, at the end, there are multiple contacts that get broken. So basically these simulations, which we did unpack, second mechanism, they determined that mechanism B is the more likely mechanism for this virucidal activity. So this work is in preparation, and the single protein work was actually published 3 years ago in Nature Materials. So here, this work we are going to submit soon, now that we finished, and show this evidence of the virucidal activity. So with that, I would also like to say a little bit about the second project that we also ran on TAC, and that is related to development of carbon nanotube-based nanosensors of molecules that are called neurotransmitters. So you may have heard of the neurotransmitter molecules. They are basically the molecules that are signaling in the brain. This is an image of how of the junction between 2 neurons in everybody's brains. So when there is some signal that is passing from one neuron to another, the way it happens is that there are these little vesicles at the edges of these neurons called synapses. There are these vesicles filled with molecules called neurotransmitters, and then these vesicles burst, and the neurotransmitter molecules diffuse around and pass on the signal. So you probably heard of these neurotransmitters, maybe not in this form, but you heard maybe of dopamine, serotonin, such molecules. So if one is craving to eat some food, you start feeling this surge of dopamine, and then when you eat it, you feel a great pleasure from it. So that would be an example of a neurotransmitter molecule, so dopamine. You can see what happens, right? This is basically a long protrusion of a neuron cell where this dopamine bursts out and passes the signal. So many diseases, neurological diseases, are directly related to some different patterns of this neurotransmission process. So there's basically a great interest in understanding how does this emission of neurotransmitter happen inside tissues between the neuron cells, and the scientists want to study it and understand it better, how this transmission happens in certain conditions, within diseased conditions versus normal conditions. If one gives a certain therapeutic, what happens to this neurotransmitter emission? And dopamine is just one of such molecules. There is serotonin, there is a histamine, there is epinephrine, norepinephrine, oxytocin. All kinds of molecules are out there that we would like to track in these types of tissues and on these types of system. But to detect them, you need to design the sensors for that. And so detection of neurotransmitter is actually pretty challenging, though, for multiple reasons. So all of this that we are interested in, it's happening at synapses. So that means everything is occurring at these small length scales at low concentrations, and everything is happening very fast as well, right? If we want to respond to something, we know how fast our brain is working. So everything is happening very fast on the molecular level as well. So if we want to design these sensors, we face quite a challenge. The molecules themselves, the neurotransmitters, they're pretty small. They're just small organic molecules which don't have some typical molecular recognition elements. And then some of them are also very similar to each other, like dopamine and epinephrine. So there could be interfering substances that can prevent the detection. And so we worked with experimentalists on the design of these sensors, and they were based on carbon nanotubes. So carbon nanotubes are these special materials that have very attractive optical properties. If one shines light on the nanotubes, then they're going to absorb the light and emit it. And the idea of these sensors is, can we use the carbon nanotubes to absorb light and emit the light, and then in this way, through the light emission, give us a signal, is this analyte like dopamine present in the system or not? That's the idea of the sensing. So basically one would hope that we can use the carbon nanotubes to give us some light emission and alert us, oh yeah, there is dopamine present. So that's the idea of the sensor design based on carbon nanotubes. Carbon nanotubes, they are hydrophobic materials. So in order to put these nanotubes into in contact with aqueous solution, with water type of solutions, you need to actually wrap them by certain polymers to solvate them. And also polymers can serve to bind the analytes, such as dopamine, if that's what we want to sense. And so these are the types of nanomaterials, nanotubes wrapped with polymers that can be used to detect presence of neurotransmitters. It was shown actually that, for example, DNA wrapped nanotubes, one can prepare these arrays of such nanotubes, place neuron cells on top of these arrays, and then you can sense, are the cells emitting dopamine or not? So such systems have been prepared, and we want to prepare new sensors which have optimized properties. They should be stable, selective, sensitive, and reliable. And in addition to sensing dopamine, we may want to sense also other molecules such as serotonin, epinephrine, etc. So as I mentioned here, the DNA, using DNAs as polymers on these nanotubes turns out to be a very, very useful polymer for this purpose. That's what's experimentally been detected. But if you know something about DNA, it is a polymer that is made of nucleotides called ATCG, which are basically just repeated small molecules covalently bound to each other. So what we want to do is we want to design such new sensors that are going to sense neurotransmitters, and we need to find appropriate DNA molecules that will lead to this, eventually leads to a strong light coming out from the nanotube. So what one needs to do is find the good DNA molecules that will lead to this strong light coming out. The way that experimentalists do it, find the sensors, is they depend on serendipity. They choose certain DNA molecules. They choose first what's going to be the polymer length of the DNA, and then what's going to be the sequence of it. And then they prepare such system, add dopamine, and measure how much does this system respond to dopamine. Then they have to do separate experiment, prepare again nanotubes wrapped by other sequence of DNAs, add another neurotransmitter, and see is it responding or not to that particular molecule. And so you can see here example of an output in experiment. Different types of DNAs were tested, wrapping the nanotubes, and then they checked, are these, let's say you have here, for example, a sequence of GA15. That just means that you have repeated pattern, like a DNA molecule of 30 nucleotides, which has a repeated pattern of a sequence, GA, 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 GA 15 times. And so one wraps nanotubes with this type of DNA, and then one checks, is this system, if we have a nanotube wrapped by a GA repeat pattern, is it going to be giving a strong light when it's exposed to dopamine, strong light emission when it's exposed to dopamine? And it is in this case. If it's a very bright image, then that's what's happening in experiment. Then yes, it's responding, the nanotube is strongly responding to dopamine. If you have this dark signal, such as here, then this sequence G15, T15, it's actually very poorly responding to tyramine. So screening approach, and in this way it works. It finds the potential sensors, but it doesn't provide a good understanding of why it works, or how would you do it better next time if you want to design a different sensor, right? So that's where we came in, right? We decided with our collaborators, let's try and see, can we do something more systematic? Our collaborators are Marquita Landry's lab at University of California, Berkeley. So they prepared these systems in the lab, and we said, let's try to do it more systematically. Let's just check if we have the same sequence and we have DNAs of different lengths, and let's check then for these DNAs of different lengths, how they're going to respond to a range of neurotransmitter molecules. And so in experiment, they checked it. We used the sequence called GT repeat. So that just means GT, 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 many times, n times, and then sequences of different lengths. So some of them were GT4, GT6, GT7, all the way up to GT30. And here we have a heat map if this particular sequence is sensing the neurotransmitter. And you can see if it's red or yellow, it's a good sensor. If it's dark blue, it's not a very good sensor. So we could see that GT6, for example, it's a good sensor for dopamine and norepinephrine in this case, and for nothing else pretty much. So GT6 turned out to be a good sensor. And then all these longer DNAs, they're not very good sensors for these neurotransmitters. So that was the first initial measurement that was done. The reason why this happens for GT6, it turned out to be that when there is no dopamine present for these GT6 wrapped nanotubes, the nanotube was very dark. It wasn't emitting a lot of light. And when one added dopamine, then it suddenly became very bright. So basically, we wanted to understand what is the difference between GT6 and GT15, GT30. What is difference on the molecular level that could be leading to such a sensing here? We are just screening the length, right, of polymer. So my student Ali, we started this project together. We didn't know what we were going to get, so we said, okay, why don't you test these and we'll model several systems and then see what happens. So my student Ali, he started modeling these long polymer DNAs versus short, and we want to see, is there a significant difference we can observe by eye in our simulations? So you can see here a nanotube wrapped by a GT15, one of those longer DNAs, and then we will contrast it with what happens on a molecular level when we have a nanotube wrapped by GT6, shorter DNA. And so if you have a nanotube wrapped by GT15, GT15 is a pretty long molecule, and it turns out to helically wrap the nanotube. When it came to the shorter GT6, we were surprised and unexpected. It was pretty unexpected for us. We had a nanotube wrapped by GT6, and what happened over time in our trajectories was that GT6 started forming the rings on the nanotubes. If you put multiple GT6 polymers, they form 3 rings, as you can see here. So this was pretty unexpected. So they were forming rings very quickly in our simulations, in our snapshots of the system. We could see this. In chemistry, we usually think, okay, this could be an accident that it happened, but if we can prove that this state of the system has a free energy, is a free energy minimum state, then we can be assured that indeed the observation is the one that is going to repeatedly happen, no matter how many times we would test such a simulation. So the next thing we did is we used some replica exchange molecular dynamic simulations, pretty computationally intensive, to calculate the free energy landscape of a system of a nanotube wrapped by GT6 DNA. And here is a free energy landscape. It's a 2-dimensional contour plot in terms of the relevant reaction coordinates in this system. And what you should pay attention to is the color of the contour plots. So you can see the color represents the free energy of the system. If you see the blue regions, dark blue, those are free energy minima for these systems. Those are the conformations that the system likes to adopt. It's a free energy minimum. And if you see red and other colors, those are like higher energy conformations, which are less likely to occur in the system. And there are 2 free energy minima. We label them by conformations 1 and 4. So if we can go back in our simulations and check, okay, what does this basin of blue structure look like? And it turned out that this region labeled by 1 corresponds to the structures that do form ring conformations. And that is a free energy minimum structure. So this plot confirms ring structure is one of the free energy minima. The system also has another free energy minimum, which corresponded to the left-handed helix. If we checked the contour plot, if we plotted it like a surface, a 3D surface, we could also check what is the barrier for the system to go from helix to ring. And it turns out to be only a kT of energy, which means it's like it takes a thermal energy for the molecule to transition from helix to ring conformations, which means that it could be happening in an experimental system all the time, this transition from helix to ring. And so, yeah, so we observe the rings. They are definitely free energy minimum states. And then the question is, why is the nanotube wrapped by these rings very dark, not emitting the light, which is what experiments observed? So to understand that, so we know now nanotubes wrapped by GT6s, they're wrapped by these rings. And the question is, why are they not emitting light? So initially they don't emit light. Once you add dopamine and it binds to the nanotube, they're going to start emitting light. So in order to understand that, what's the effect of these rings on the light emission, we need to just a little bit mention what's the mechanism of this light emission. It's based on the nanotube. When it absorbs light in a spectroscopy experiment, it forms this state called an exciton state. It's a state where you have an electron and a negatively charged electron and a positively charged hole coupled to each other and diffusing through the nanotube surface. So we were thinking, okay, if you create an exciton state, electron and a hole, then this exciton, this electron and hole, should be interacting with the nanotube environment to these rings, right? And their electron and hole, they're these electrostatics that should be of importance in such a system. So the way the exciton emits light is that the exciton has to annihilate, it just needs to disappear, go back to ground state, and it either emits light or it goes, or it doesn't emit light. There are 2 ways it can relax down. So we were then wondering, okay, so we have electron and hole, it's probably electrostatically interacting with the environment. And the environment of our GT6 nanotube are those rings. That's what we established with our classical simulations. So we were wondering, okay, what is happening to the electrostatics around our nanotubes that are wrapped either by GT15, the long DNAs, or GT6s, the ring-like DNAs. And the electrostatic potential at the nanotube surfaces, due to the DNAs wrapping them, turned out to be either random, kind of random potentials. You see, there is no pattern here that we observed for the long DNAs. But when it came to GT6 DNAs, these rings, the electrostatic potential in which this exciton was living in turned out to be periodic. So you can see here, electrostatic potential, we tracked it along the nanotube length. And when you have the rings there, the electrostatic potential was periodic. That's the potential in which exciton existed and it needed to relax back. So seeing this difference of the periodic potential for the GT6s, we were wondering what effect could that have on electron and the hole that need to recombine and go back to ground state. And we designed a simple physics-based models for what happens to electron and hole wave functions inside periodic potentials. Very simple. So this is like some graduate physics model that the students, the starting students in the physics department do, chronic penny potential. But we're chemists, so for us it was complex, but we anyways prepared these electron and hole wave functions inside very simple periodic potentials. And what we find with these physics-based models is that if you have a periodic potential, the electron and hole wave functions will avoid each other. And that will result in a significant cancellation of their overlap, which will affect the oscillator strength. And eventually, the small oscillator strengths are going to lead to low light emission upon exciton relaxation. So basically, we were able to show with a simple physics-based model that if there is a periodic potential present where the exciton exists, then we are expecting the nanotube not to emit light. So that's the conclusion coming out from classical modeling. We observed these unusual rings. We kind of pushed that down to this physics-based model, and we saw the effect that these nanotubes should be dark nanotubes. And that's what the experiment showed. So we could explain that. And then we also wanted to check, so what does the dopamine do when it binds to the nanotube? Here is dopamine binding to these ring-wrapped nanotubes. And we simulated for a pretty long time of about 2.5 microseconds. So dopamine didn't do much. It bound to the nanotube surface over first 1.5 microsecond, but eventually the binding of dopamine led to the distortion of the rings. So distortion of these periodic potentials. So we think that dopamine insertion, it can disrupt the ordered structures at the nanotube surface, and then that could lead to radiative relaxation, meaning light emission of the nanotube. So seeing these rings was quite unusual for us. So we basically did a follow-up study also with the help of TAC, because we did a lot of screening. We did a follow-up study to check are other sequences also forming rings if they have the correct length. And so we screened a lot of the sequences and found that ring formation on carbon nanotubes is definitely sequence dependent. So some sequences, you can see if they're in green, they have high propensity to form rings. If they're in white, in light green, some of them, they don't form rings. So through screening, we observed that. We also characterize the free energies for multiple sequences. So for example, GT6, we already showed that it can exist in ring and helical states. Another sequence, C12, it's just a 12C nucleotides DNA, it very strongly likes to form rings. That is the free energy minimum for this type of sequence. Sequence like T12, for example, it doesn't like to form rings at all. It actually likes to form the loops. So it is highly sequence dependent. The conformation is highly sequence dependent. Through analysis of these sequences, we also observed what drives the ring conformations. If the nucleotides, if the bases are forming special hydrogen bonding, then that's going to lead to the ring formation. Also, some sequences could have higher probability of having sodium ions inserted close to the backbone of the DNA molecule. We also observed that, and that should be leading the ring formation, such as in these sequences. So this was also published last year. This exploration of different sequence dependence on the ring probability of the ring formation of nanotubes. We could also, the experimentalists also checked, we screened, is this favored conformation of the DNAs, is this going to be affecting or having some correlation with the kinetic stability of these DNAs on nanotubes? That's what the experimentalists measured, the kinetic stability of these DNAs on the nanotubes. And we found basically that kinetic stability depends on the conformation and also on the carbon nanotube binding affinity, which we showed correlations for together with them. But our work was basically screening all these sequences computationally. In a more recent work, we are trying to find more systematic ways to identify good DNA sequences that recognize the neurotransmitter we want to detect, we want to optically sense. So two students in my group, Payam and Juan, they're working on this project where we are taking different sequences that have been tested in experiment that sense, in this case, serotonin neurotransmitter, and we are training machine and deep learning models based on these sequences and their sensing ability to try and predict new sequences that will be better sensors of serotonin. So this is a recent project which we also want to use. We use Frontera or Stampede for, but we still didn't get to that at that point. But we have had actually quite a nice success. We trained some models. We gave the predicted sequences to experimentalists, and then they tested these validation sequences and found actually in total now it's like 5 better sensors of serotonin thanks to the models that we trained. So it's quite promising. We want to apply to these machine learning and deep learning models to detection of other neurotransmitters if we can find better systems. So with that, that kind of ends my talk. I would like to acknowledge my collaborators, who you can see there are a lot of experimentalists and another theoretician who worked together with us to get these results. My group, all of the students contributed to the projects I presented, and our funding from NIH and NSF for the 2 projects, and of course without the computation time from TAC and other, we would not be able to do this work. So I'd like to thank you too. And if there are any questions, I would be happy to answer them.
Great. Thank you so much. That was really impressive. Those projects and the papers. Yeah, very, very impressive. I guess what I want to say real quick is if anybody has any questions, please maybe hit the raise your hand emoji thing or unmute yourself, and please feel free to just ask or post in chat. While questions come in, I'm going to ask a quick one first. I'm always really impressed when I see these like really complex atomistic systems that you simulated, and I don't know whether or not, is there experimental models that you are simulating from or whatever, but so I guess that's my question is when I see something like a carbon nanotube wrapped in DNA, or this like quadrimer of viral receptor proteins in a nanoparticle with all these complex things on it, like how do you guys build these systems? Are you building them by hand? Do you use some tools to build them, or are they from X-ray or similar?
Yeah, so many of these systems, especially when it comes to biomolecules, one can crystallize them and obtain the crystal structures for. The capsid, which I was showing for the viruses, they were actually coming from cryo-electron microscopy structures. So yeah, for biomolecule, there is a lot of structural data. This is where we start from. For materials, we have to just write our own codes to build them. Usually one also knows what is the lattice of the material or what is the nanotube looking, but if there is no code available to build these, we have to make them on our own. So that's the way it works. And typically one can kind of predict what happens with materials. You know what the gold piece of gold looks like, and one just has to build the ligands or things like that. So it's a mix and match. Whatever there is a structure for, we just download it and use it. If there isn't, we build our own. So we have to use our coding. Yeah.
I'm going to pose a question from chat and then I'll go to Ethan. So this is regarding your first project. So those particles, would they be helpful for people infected already with COVID, for example, or and also do they have side effects for people who are not infected?
Yeah. So the envisioned usage of those nanoparticles by experimentalists, it was basically to more of like a topical cream, which one could apply, and then it would lead to the destruction of the viruses. For some of the viruses, right, like papilloma virus or or such things. It wasn't really the ingestion of the material that was intended. However, the nanoparticle materials, they were gold and iron oxide, which are non-toxic materials. So they were paying attention in experiment. Let's make this, but let's make it for materials that one can actually apply on the skin topically, so that there is no side effects. So yeah, it's like broad spectrum, non-toxic. Those are the goals for the research.
So would it be helpful if someone takes them before getting a COVID? So if they stay in the body for a long time, so they can act as the antibodies, right?
Yeah, that would be, so this was not designed for COVID. This basically, the first paper that was done was, I think, in 2018. So we did it a long time ago. It was, as I said, just intended, let's find the materials which destroy the viruses. And for COVID, I don't think it would be like something that you would ingest after you get infected. Yeah.
Thank you.
Mm-hmm.
Maybe it could be a coating for like the inside of a mask, double the effectiveness of a mask or something.
Yeah, I think, I've seen actually, that's a good point. Yeah, I've seen some masks being sold like virucidal shield. Yeah.
Ethan, did you want to unmute?
Yeah, sure. Sorry about any background noise too, but yeah, so I really liked the talk. I thought it was cool how you looked into sequence dependence here. My question is, it's pretty well known that DNA, like especially GC sequences, are dependent on salt conditions. Did you check, or do you have any interest in checking the dimension of checking different salt conditions for sequence dependence there?
Yeah, that's a good question. I mean, from physical chemistry perspective, which is what we are interested in such things because we want to know fundamentally what's driving these conformations and sequence dependence. So we are interested in such questions, and you can see here when we were screening, we were looking at 1 nanomolar salt rather than 0.2 molar salts. So from that perspective, yes, we are interested. A lot of the experimental applications, they want to put these sensors inside tissues, inside, like, let's say, brain slices. So those are physiological conditions. So the salt concentration would probably not make would basically, varying the salt concentration would not be of such an interest for application usually, typically, if one is testing in tissues and at interfaces of cells. But for us, yes, we are interested, and we were testing some of these things. Yeah.
Cool, thank you.
Thank you.
Any other questions? I might ask just one more quick one out of curiosity. I guess for both projects, I'm more interested in the first, the nanoparticle. But what sort of force fields did you use, and like what simulation tools? Was it coarse grained? So just like some of the details I'm curious about.
Yeah, so I basically came from a group which I did postdoc in a group that developed this NAMD software. So I'm kind of partial to NAMD. It's just like it has been developed to be working really well on these highly parallelizable platforms. parallelized platforms. So when we have these systems of 1.5 million atoms, we like to use codes that have been optimized for that. And NAMD just traditionally. Charm force fields, not for this project, but for others, we do use coarse graining with GROMACS and Martini. We were thinking of doing it for this project because you could get much longer time scales and a lot more dynamics. So, but we didn't do it for this project yet. But yes, that's what we use. So, yeah.
Well, if there are no other questions, I'd like to thank you one more time for joining us in the seminar, and thank everyone else for their attendance and participation. And I look forward to seeing everyone here at the symposium in September.
Thank you very much.
Thank you again. Take care, everybody.
Bye.